Well, good evening, everyone, and thank you to the Natural History Museum, and especially Lisa Thompson for that wonderful introduction, and for inviting me to speak here tonight, and for those of you who have come to join me in this discussion. So, as Lisa already pointed out, this is one lecture in a series of lectures on what it is to be human. So, I assume that each and every one of you here has asked yourself this very question, and I'm assuming that a lot of you come up with answers like our greater capacity for cognitive thinking makes us differentiated from other mammalian species, or perhaps it's our ability to explore our environments and undergo scientific endeavors. But I guarantee that none of you, or maybe one or two of you, but most of you don't think of the bacteria that live on your body as being important for who you are as a person and being important for shaping the human species in general. So tonight I'm going to talk about the commensal bacterial species that live on your body that we collectively refer to as the microbiota. And we're going to talk about how these microbes are important for immune system development, for metabolism, and recent science is showing it's even important for our behavior. And I'm hoping that by the end of this lecture, that many of you will become to appreciate how important these commensal bacterial species are to our health and our susceptibility to disease, and that we'll be able to redefine who we are as humans, at least to include the commensal species that live on our bodies. So, oops, I have to stay close to this microphone. So we live in a microbial world. In fact, bacteria predominate life on Earth, and you can find these organisms pretty much in every niche on the planet. They evolve very quickly, and they can adapt very quickly to every environment. So you can find bacterial organisms in very benign environments, but you can also find them in the harshest of environments, including the hottest springs, or even in the Antarctic. So the human species had to evolve in the presence of these microorganisms. Now, oftentimes when I talk to people about bacteria, they instantly throw their hands up and they think, oh, bacteria are bad. And this is in fact ingrained within us at a very young age. So this is a picture of my 2.5-year-old daughter, Alexa. And one day I was sitting at home reading a microbiology magazine, and of course in a microbiology magazine there's lots of pictures of petri dishes with bacteria on them. And she came up to me and she pointed to this petri dish and she said, "Mom, what is this?" And I said, "Oh, babe, it's just a dish with bacteria." And she looked at me in horror, her eyes wide open, and she said, "Oh, that's bad. Bacteria are bad." So even a 2.5-year-old thinks that bacteria are bad, and she's being raised by a mom who thinks that most bacteria are good. So I've done something wrong here. And hopefully I'll convince you that most bacteria are good as well tonight. So we have to ask the question, are all bacteria in fact bad? Most of the bacteria on this planet don't cause us harm, and the bacteria that live on our bodies, our immune system doesn't even respond to, and they don't make us sick. So as I've already alluded to, each and every one of us in this room is home to a vast consortium of commensal bacteria that take up residence in almost all environmentally exposed surfaces of our body. And in fact, these organisms are so numerically prominent that they outnumber our own cells by an order of magnitude. So what that means is for every cell on your body, every human cell, there are 10 bacterial cells as well. So, for all intensive purposes, each and every one of us is more bacterial than we are human. So, if you remember not too long ago, there was a big push to identify all the genes that make us human via the Human Genome Project. And in fact, they identified all the 23,000 genes that make us human. Now, when you compare that to the number of genes that are actually found in the microbiota, these organisms that live on our body, you can see that there is an astounding number of genes. And actually, this number is estimated to be up to 4 million different genes. So this is a huge variation of gene products, including our own genome, that make up who we are. So as I mentioned earlier, most of these microbes are found on environmentally exposed surfaces of our body. So this includes our mouth, it includes the lungs, our skin, the urogenital tract, as well as the gastrointestinal tract. And the gastrointestinal tract includes the stomach, small and large intestine. Now, if you look within each of these bubbles that represents a particular area of your body, you'll see that there are a lot of branches. And these branches represent particular species that are found on the body. And just to give you a feeling for how little we actually know about these microbial species, is a red little dot has been placed on each branch if any scientist has identified the organism and some function has been attributed to these particular organisms. And what you'll notice is that there's very few red dots within many of these areas, which means that we have a lot of work to do insofar as identifying what these organisms on our body are doing. What you will notice, however, is that there are quite a few bacteria that have been identified that reside within the gastrointestinal tract. And this is largely because the greatest density and diversity of the organisms that live on our body are found within the gastrointestinal tract. And in fact, as you move from the stomach through the small intestine towards the large intestine, this density and diversity increases. So the largest burden of bacteria on our body are actually found within the large intestine. Now, this is a very interesting place to study host-commensal interactions. Because I actually consider myself an immunologist as opposed to a microbiologist. And as an immunologist, I've been taught that our immune system is designed to recognize foreign entities and get rid of them. And actually, 60 to 70% of our immune system is actually found within the intestinal tract, coincident with our largest burden of bacteria. So this is a very interesting place to try to understand why our immune system tolerates these bacteria and how they tolerate it. And we'll actually come back to that a little bit later in the talk. So the study of the microbes that live on our body has been something that's been interested in for centuries. Scientists have been studying. But the real momentum in this field was gained when Dr. David Strachan proposed what is now termed the Hygiene Hypothesis. And he proposed this hypothesis in response to a series of data, which I'm showing here. So if you guys focus your attention to the graph on the left, you'll see that this graph shows from a period of 1950 to the year 2000 that pathogenic infections or infectious diseases, such as diseases of rheumatic fever, Hep A, or tuberculosis, that over this time frame that they undergo a rapid decline in their incidence. And this is not surprising because around this time, vaccinations and heavy antibiotic use, as well as increased sanitation, were found in westernized civilizations. Now, if you look to this graph on the right-hand side, over the same period, in the same exact places, you find that there is a startling increase in the incidence of autoimmune diseases. So these are diseases where your immune system attacks your own cells. So diseases such as multiple sclerosis, where your immune system causes a demyelinating disease and actually attacks the nerve fibers. Inflammatory bowel disease, such as Crohn's disease, where your body no longer tolerates your commensal bacteria within your gastrointestinal tract, as well as type 1 diabetes, asthma, and allergies. So David Strachan proposed that because antibiotics that we were using to cure all of these other pathogenic infections are not discriminatory between organisms that are pathogenic and our commensal organisms, that what we were doing is we were influencing our relationship with these commensal communities, and in so doing, we were predisposing ourselves to autoimmunity. So this was proposed in the late 80s, and since then, this field has really gained some momentum. So one of the most important techniques that has been developed in the last decade or so has really allowed us to identify the organisms that live on our body. So most of the organisms, especially within the gastrointestinal tract, are unculturable. And this is largely due to the amount of oxygen that's present. And many of these bacteria can only grow in anaerobic or without oxygen environments. So it was very difficult to grow up these organisms within the laboratory and study what they do and who they are. So with the advent of next-gen sequencing, which I'm not going to get into the details of that now, has really allowed us to be able to take fecal samples from humans and from animals and really identify the different organisms that are present on our body. And also, we can determine what genes these organisms are expressing, so we can infer some function. And this is a big part of the initiative by our government to understand the microbiome through the HMP, or the Human Microbiome Project. So recently, this consortium has published a lot of their findings to identify what it is these organisms are doing on our bodies. So some of their findings I'm going to present to you in the next couple of slides. And what they have found is that our microbiota is much like a fingerprint. So no two people in this audience have the same microbiota. So all of the different species on our body are very different. But what they're finding is that the functional capacity of these organisms can be redundant. So different species can have the same function and provide you the same benefits, so to speak. So I'm going to show you quite a few of these plots. So I'm just going to take you through this one to demonstrate some of the findings that have been published for the Human Microbiome Project. So the way that they do this, they represent this data, is that each one of these little dots here represents a single person, but it represents all of the different microbial communities on their body. And the way that you read these particular plots is if you see them clustering together like this, this means that the microbial communities are very much alike. However, if you see differentiation of these dots, that means that their microbial communities are very different. So in performing these sequencing techniques, they sample thousands of different healthy individuals. And what they have found is that the microbial communities all over our body are very different. So the community within our mouth is different from the community on our skin, which is different than the community within our gastrointestinal tract. And this is not altogether surprising, given that each of these habitats has different things that the bacteria need to deal with. So this is shown here where the pink is the microbiota of the skin from what looks like hundreds of different individuals. This would be the microbiota of the intestines from different individuals, and then the microbiota of the mouth. So you can see that since they're segregating very discreetly, that these microbial communities are very distinct. Now, when you look at the microbial communities between males and females, what you'll find is something very different. So up here on the right-hand side is the same plot, but what they've done is they've highlighted all the females within this plot in red, and they've highlighted all the males in blue. And what you can see is that there is no segregation of these particular dots, which means that the microbial communities between males and females, despite our incredible differences, our microbial communities are fairly the same. So it seems that one of the biggest differences is that the body habitat is very important for what types of microbes reside there. So I didn't want to get into too much nomenclature about the bacteria that live on your body, but I thought I might highlight some of the bacteria that you might have encountered or heard about in your lifetime. So there are over 100 different phyla of bacteria. So phyla is the highest classification of bacterial communities, and within each phyla you can have thousands of different species. And you can find these all over the world. But what's interesting is that when you look at the bacteria that live on our bodies, they're very specific. You can't find just any old bacteria that lives there. They seem to be highly specialized. And in fact, what you see is a predominance of the Firmicutes and the Bacteroides on the different locations of our bodies. Now, one organism that I'm going to talk to you about today is a member of one of these dominant phyla of our body, and that's Bacteroides Fragilis, and we've identified it as a beneficial microbial species. Some of the other organisms that you might have heard of are members of the dominant Firmicutes phyla, and that's Clostridium and Enterococcus species, which most people have heard about them as being bad, but we actually have some commensal species. But one of the more interesting ones are the Lactobacillus species. So how many of you guys eat yogurt? So a lot of times this is the organism that is found in your yogurt. And there's a lot of different strains, and the literature is showing that this is a very good bacteria. So this is a prominent member of the human commensal microbiota. Other organisms you've likely heard about are E. coli. So again, not all E. coli are bad. There's actually some beneficial commensal species. And Bifidobacteria is another member. It's not as numerically prominent as, say, the Lactobacillus, but this is another bacteria that is featured in some of the yogurts like Activia, as well as some over-the-counter probiotic pills that many people these days are taking. So this is just an introduction to some of the members of your microbiota, but there's about 1,000 different other species that we're trying to identify. So one question that I always get is, how stable is the microbiota, and does timing matter as far as colonization is concerned? This data was recently published in Nature, and it's again part of this human microbiome project. And if you look at this bottom axis here, you'll see that they've sampled many individuals from many different countries of different ages. And what they've put on the y-axis here is a difference between the microbial communities of adults and of children. And what you basically see is that through ages 0 to 3, there's a vast fluctuation in the microbial communities. And that the microbial communities in these very young children look nothing like the microbial communities in these adults. But after that 3rd year, it looks like the microbiota begins to stabilize and look more like an adult microbiota. So it's thought that your environmental exposure during those first 3 years is very important in shaping your microbiota and who colonizes you. And this is especially important in young children who have to take antibiotics, because now you're obliterating any organisms that you're colonized with. So it's during this first 3 years that seems to be very important. So what are the things that can influence our microbiota? So I've already talked to you about antibiotics, and we'll talk a little bit more about this in a minute, but there's other things that can influence this as well. So things like your genetics. Not that you can do anything about what your parents gave you, but this is very important in deciphering who can colonize and who cannot. Additional things that you can control are your diet and your stress levels. All of these things influence the type of microbiota that we're colonized with. Moreover, we'll talk about this in the next slide, is during those first 3 years of life, I mentioned it's very important what you get exposed to. Well, this early colonization, so birth in various hospitals, whether it's a dirty hospital or a clean hospital or cesarean versus a vaginal birth, all of these things can influence the type of microbes that you come into contact with. So one of the interesting things that came out of the Human Microbiome Project is a study that showed that how babies are born influences what organisms they're colonized with. After you see the data, you realize, of course, that makes sense. But what they did is they sampled mothers and their babies, and you can see that in green they sampled the mother's mouth, her vagina is in red, and the skin is in dark blue. And then they also sampled the baby's microbiota, and they sectioned it off into a vaginal delivery, which is in pink, and a C-section, which is in light blue. And I think what you can readily appreciate is that the microbiota of the baby that was born via C-section is very much like the skin of the mother, and in fact, it's dominated by staph. Moreover, if you look at the babies who underwent a vaginal birth, it looks very much like their mother's vaginal flora, and this is dominated by the Lactobacillus and Prevotella species. So this is important because it's becoming increasingly clear that babies that are born via C-section, they tend to struggle with staph infections. And they think that one of the reasons this might be is because their first exposure to bacteria is through their mother's skin, which has quite a bit of staph. So it seems that how babies are born influences their first exposure to commensal bacteria. So not surprisingly, how babies are fed also influences these microbial communities. So if you look at babies that are breastfed, they're shown in pink, versus babies that are formula-fed, you see that there's a very discrete separation. And in fact, what they have shown is that these babies that are formula-fed, they tend to have a greater diversity of beneficial bacteria. And in fact, this is correlated with decreases in development of autoimmunity later in life. Now, this is not any pressure for women to breastfeed their kids, but this is important for scientists to know, because what could be done is to identify what bacterial species are being provided by the mother. And it would be great if we could begin to put those bacterial species into formula so that babies get this exposure early on in life. So one of the most important things in my mind is what happens to the commensal microbiota when you take antibiotics. So it's not surprising that the commensal microbiota is indeed influenced, but here's a little bit of data to prove that point. So what's shown on top is a pie diagram which represents the diversity of a given microbiota in an individual prior to antibiotic treatment. So you can see that there's a lot of colors there, which means that there's a lot of organisms. And it's thought that the more diversity of organisms that you have, that the more beneficial and more healthy that you are. So C. difficile, or Clostridium difficile, is an organism that is associated with traveler's diarrhea. So let's say you go into your doctor and you have a C. difficile infection, and they prescribe you antibiotics. So two of the most popular antibiotics are vancomycin and metronidazole. And what happens is you do, in fact, clear your infection, as you can see from this cartoon diagram of the diversity of organisms. Clostridium is represented as these red rods. So you can completely ablate those particular bacteria. But then look what happens to the rest of the microorganisms. You can see that different antibiotics decrease this diversity very rapidly, and now you're left with only a few organisms that you hope will be beneficial. So I think the major question here is, well, can these commensal microorganisms recover after antibiotics? So those studies have been performed in animal models, and they're currently being performed in human populations, but I'm going to show you the animal data here. So what they've done here is they have listed a bunch of commensal species that live on the body, and this is a pretty basic picture where they've taken a look at the organisms during antibiotic treatment, and they've compared them to the starting place. And they've looked 3 weeks after the animals were off of the antibiotics to see what happened to these organisms. Now, if there are absolutely no changes to the commensal microbiota, both of these blocks would be completely black. So black indicates that there's no change. If you see a green box, that means that that particular species was decreased over 100-fold. Whereas if you see a red box, that means that particular species increased 100-fold. So I'm not going to go down this entire list, but what you can see is that during antibiotic treatment, there are vast changes in the commensal microbes. You see increases in Enterobacteriaceae, and you see decreases in the Bacteroides. So E. coli is part of this family right here. You also see increases in other not-so-good bugs, including Enterococcus, as well as Streptococcus. So that wouldn't be so bad if you were able to recover your commensal organisms. However, it's looking like that even 3 weeks after the antibiotic has cleared the system, that you have a stable loss of some of the beneficial commensal organisms. So what you can see is that all 3 different antibiotics, ampicillin, vancomycin, and metronidazole, result in a stable loss of the Bacteroides phylum. And one of these causes an overgrowth of this E. coli family. Additionally, you also see an overgrowth of the Enterococci, and you see a loss of these Lactobacillus species. So it seems that antibiotics do cause permanent effects to the microorganisms. So in the future, it would be best, once we figure out what organisms are important, to couple antibiotic treatment with some sort of probiotic treatment afterwards so that we can replace some of these beneficial species. Now, what might be troubling is we should really be asking the question, so if you change your microbial communities permanently, does that mean anything for your health? And what we're starting to find out is that treatment with antibiotics early in life predisposes you to diseases such as IBD. So I'm just showing you a correlation study that was performed not too long ago, where they went back and they looked at people who had inflammatory bowel disease, and they asked how many antibiotic treatments did they undergo very early on in life. And what you can see is people who didn't have any antibiotics have a very low susceptibility to IBD, whereas as you increase the number of antibiotic treatments you had as a child, your susceptibility to getting IBD goes up. So this is a piece of correlative data that suggests that antibiotic treatment early in life might predispose you to these particular diseases, which is consistent with David Strachan's hypothesis. So I briefly wanted to comment, so I wanted to stop showing you plots about changes in microbial communities, because at the end of the day, it doesn't mean much. There might be changes and they might cause disease, or they might just be a consequence of disease. So I did want to point out that when people do these massive sequencing experiments, they compare people who have diseases like multiple sclerosis, even obesity, diabetes, even autism, during pregnancy, arthritis, and IBD. When you go and you take the microbiota from those people and you compare it to healthy individuals, you see huge changes in each of these people's microbiota. So again, we have to ask the question, is this cause or consequence? Is the disease driving these changes, or is it the microbial communities that are driving these changes? So the real way that we can do this is we can do this in animal models. So there are a couple of animal models I just wanted to point out that scientists are using to try to better understand how the microbiota influences our health and disease. Most scientists tend to focus on the animal species mice or rats. And this is because these animals recapitulate in many ways human disease. And their microbiota looks very much like a human microbiota. Whereas if you look at other organisms such as fish or flies, their microbiota is dominated by very different species. And they have very different immune system function. So we'll talk a little bit about what's known in mouse models for these various diseases. So one thing that Lisa actually mentioned is that one of the best demonstrations of how important the microbiota is, I think, comes from animal models where we make these animals germ-free and then we compare them to animals that have lots of different microbes. So what we do is we take these germ-free mice, and they live in these little plastic bubbles, and they are never exposed to any type of bacteria or virus or parasite that we can assay. And when you compare the development of these animals to their conventionally colonized counterparts, you see that these germ-free mice have a massive number of defects. In fact, these animals have nutritional differences, so they have to take in more food to maintain the same amount of body weight. Their liver is smaller, the heart is smaller, they have increased cholesterol. And then if you look at any immunological parameter, they're deficient in almost everything. So they have less immune cells, they have less antibodies, their intestine even morphologically looks different. More importantly, when you challenge any of these germ-free animals with your favorite pathogen, your favorite nasty bug, I don't know if any of us have favorites, but every time these animals are more susceptible to disease, they can't fight off the infection and they go on to die. So what this tells us is that the microbiota is important in so many aspects of our health and development, from helping us to fight off various nasty bugs to even processing our food, as well as the development of our immune system. So I wanted to go over 4 examples. I don't know how much time I have, but I wanted to go over very briefly 4 examples of how the microbiota impacts these different aspects of the mammalian lifestyle, so to speak. So the first one that we're going to talk about is animal models of obesity. So most people appreciate the fact that the microbes that live in our intestines help us to digest food that we don't have the capacity to digest. So a group of people at WashU wanted to ask, does the microbiota influence what we eat, how we process it, and does it influence our ability to gain weight? So women always like this question. So there are mouse models of obesity. There are many mouse models, and I'm just pointing out this one. So over here on our right is a regular mouse. It weighs about 25 grams. You can see that to its left is a mouse that is overtly obese. It's about 5 times as big as this wild-type counterpart. And the reason that this animal is so large is because the gene within this animal, leptin, has been mutated. And leptin is a hormone that regulates your appetite. So this is a hormone that signals to your brain that you're satisfied with your meal, so you better stop eating. So with the lack of this gene, this animal is never satisfied and continues to eat and eat and eat. So this is one reason why he's rotund. So this group at WashU, Jeff Gordon's group, wanted to ask, now are these microbial communities different? And functionally speaking, do these microbial communities contribute to this animal's weight gain? So they first looked at the structure of the microbial communities, and if you just focus in on the firmicutes and the Bacteroides, the obese animals are shown in blue, the lean animals are shown in green, and what you can see is that there are huge differences. That the obese animals have a large increase in the firmicutes, whereas the lean animals have more Bacteroides, so they have a reciprocal microbial community. And what's really interesting is that you're able to extract the feces, and you can measure how much energy is within the feces of these animals. And what they consistently found is that there was more energy within the lean individuals and less energy within the feces of the obese individuals. And what they interpreted that to mean is that these obese individuals were actually extracting more energy out of their food when compared to the lean individuals. But that's not the important experiment. The important experiment is, do these microbial communities actually induce weight gain? So this is a very classic microbiota experiment. So you take lean, germ-free animals, so both of these animals are wild type, they both have this leptin gene, and you perform a fecal transplant. So you take a transplant from a lean mouse, and you put it into this germ-free lean mouse. Alternatively, you take the microbiota from an obese mouse, and you transplant it into a lean mouse. And then you ask, a couple weeks later, what happens? Do these animals gain weight? So what do you think? Do they gain weight? I wouldn't be here telling you this if they didn't, right? So what happens is the animals that received the lean microbiota stayed lean. They stayed the same size as they were before. But within weeks, these animals that received the obese microbiota, they gained a dramatic amount of weight. And in fact, if you look at the amount of weight they gained, they became 2 times as big as their lean counterpart. So that indicates that the microbial communities that were shaped in this leptin-deficient mouse actually were extracting more energy and causing this animal to gain weight. So now where we're at is trying to identify what these organisms are. Who is responsible for extracting more energy? And can we change this composition of the microbiota so that people can extract less energy? So the implication would be, can we lose weight using a probiotic? So that would be a dream for a lot of women, right? So that's interesting, but altogether not surprising, because we know that the microbiota does this. But what about the microbiota that doesn't influence immune system development and inflammatory bowel disease? So, as I mentioned earlier, this is projecting horribly, is that 60 to 70% of our immune system is found within the gastrointestinal tract. And what I'm showing you here is a cross-section of a mouse intestine. So the mouse intestinal tissue is shown in blue, and this is the lumen of the intestine, And what we've done is we've labeled the intestine with a red fluorophore that labels the bacteria in particular. Now, what you should see, but can't, is that this lumen of the intestine is chock full of red bacteria. And one thing that you can appreciate from this is not only the numerical prominence of these bacteria within the intestine, but you can also see, or maybe not, but I'll tell you what you should see, is that they're in very close association to the host tissue. So as I mentioned before, the immune system is ready and poised here to recognize foreign entities. But my laboratory is really interested in understanding how come the bacteria are tolerated by the host. Why aren't responses being mounted? Because in the case of inflammatory bowel disease, this loss of tolerance is what drives disease. So we feel like if we can understand the mechanisms by which tolerance is induced, that we can begin to come up with novel therapies for disease. So one thing that we got interested in is there were a couple of reports that came out around this time that demonstrated that people who had IBD were lacking in a lot of different bacterial species. And one of these bacterial species was Bacteroides Fragilis. So I think I'm going on time? No? Oh. One of these bacterial species would be Fragilis. So we became interested in whether or not we could actually protect different animal models of disease. And we became interested in looking at a particular polysaccharide that this organism expresses called polysaccharide A, or PSA. And we knew that it was interesting because we could treat germ-free mice with this particular polysaccharide, purify it from the bacteria, and it could induce maturation of the immune system. So I'll quickly go through this because I know you guys will be interested in the behavior stuff. We basically orally treated the animals with a control or with this polysaccharide. We would induce inflammatory bowel disease, and then we'd look at the pathology of the animal. And what we found is that I'm again showing you cross-sections of the intestine of these mice, and that they're all at the same magnification. And you can appreciate that this particular slide, or this section of the intestine, is much thicker than the healthy intestine over here. You have a lot of epithelial cells that are proliferating, and you have a lot of immune infiltrates. But when you take that purified polysaccharide from the commensal bacteria and you treat these animals with this organism or with this polysaccharide, you can see that you can almost completely protect these animals from inflammatory bowel disease. So the implications for these studies are that there are thousands of different species of bacteria within our intestine, and if we can identify the mechanisms by which they can influence our immune system development and protect us from disease, we can get our hands on a whole array of novel bacterial-derived therapeutics that could be used for diseases that currently have no cure. So the thing that I find really, really fascinating, and this is not my particular area of research, but it's pretty new and it's cutting edge, is there's a group of people who are interested in how gut microbiota influence our behavior and the brain. So they use these really interesting behavior tests to get at anxiety levels in various animals. So what they're using here is an elevated plus maze test, which is for anxiety. So mice do not like to be up high, and they also like to be in closed spaces because that keeps them safe. So if you keep them on here long enough, they'll eventually venture out into the open arms of this particular maze. And what they can measure is the time spent on these open arms and the number of entries where these mice come out. And the more time that they spend out here, the less anxiety that they have. So in looking at how the microbiota shape anxiety, these investigators took these germ-free mice versus colonized mice. So the germ-free mice are in black, the colonized mice are in gray. What they found is that germ-free mice actually come out into the open arms more often, and they spend more time out there. So they concluded that without the microbiota, these organisms are, or these mice, I should say, are less anxious. So that's actually probably not good if you're a field mouse, because you need to have an ability to sense predation and be a little bit scared. So potentially the microbiota has evolved to help these organisms protect themselves. So since I didn't have time to go over all of the different things that they found, germ-free mice also have elevated levels of serotonin. They actually have a reduced capacity to remember things, so the microbiota is important for helping them to remember. They also have an exaggerated stress response, and they have altered gene expression within the brain. And this is all being influenced by microbes that live within the gut. So this is an exciting new field of study to understand how these microbes can actually affect our behavior. So the final thing I wanted to talk about, which I won't go over too much, is that there is some data in flies to suggest that the microbial communities that live in our bodies can actually influence who you pick as a mate. I know that we're not flies, but you never know. So basically what they found is that the flies that had completely different microbial communities would not mate with each other. They mated with the flies that had the same microbial communities. And then when they made these mice germ-free, when they got rid of their microbial communities, everyone was promiscuous and would mate with anybody. So the implication here is that maybe in the future you'll no longer send in a personality profile in Match.com to find your mate, but rather you'll send a fecal sample, and they'll look at your microbiota. So anyway, I will end by hoping that everyone here can appreciate that we are covered in microorganisms. These microorganisms have a profound impact on our health and our disease, and potentially who we are and how we've evolved as a human species. So perhaps you will agree with me that we can redefine ourselves to include the microbial communities that live on our bodies. So with that, I'll thank you very much for your attention.